Hi everyone, welcome back to the channel. This is Dr. Asi, and today's topic is BSC 1010 General Biology 1 Lab and Module 3 review. So there is a study guide for this module available for you, and I'm going to go over some key points. So let's get started. So in Module 3, we have labs 8, 9, 11, and 13. 8, 9, 11, 13, four labs. 8, 9 is mitosis, meiosis. 11 is chi-square test, and 13 is Mendelian genetics. And next test is going to cover just these four labs. So make sure you review these, look at your book, your notes, lab notes, and also study guides, and also PowerPoint slides of the lab available in Canvas shell of your lab. So let's get started. Okay, so maybe we start with one of these tests, which is called chi-square test, which is going to be a big, large problem that you need to actually calculate on the test, which we are going to have a table, uh, basically have phenotypes, column, observed numbers, which will be given to you, expected, observed minus expected, observed minus expected square, observed minus expected square divided by expected. So make sure that you can actually work this chi-square problem, find chi-square calculated. You want to compare it to the chi-square table value, which will be given to you. You want your chi-square calculated value smaller than table value to accept the hypothesis. If you accept the hypothesis, that means you basically whatever ratio you're testing, you accepted that your genetic situation is following that specific ratio, and your conclusion is that basically in that problem. So um, then the Mendelian genetic. This is directly related to Mendelian genetics, right? So um, Mendelian genetics have vocabulary that you should be studying, like what is crossing over. Allele. Allele is like a different version of a gene, like blue eye color, which is an allele of gene eye color. Diploid, haploid, fertilization, gamete, recombination, genotype, phenotype, homologous chromosomes, hybrid. So make sure you study these. They are also listed here with their definitions. And basic genetics, right? You're crossing, and in order to cross something, you should also be able to get the gametes from phenotype of parents, or if you can gametes given to you, you should be able to predict the parent's genotype, right? And basic Mendel's genetics. Make sure you also study Punnett square, dominant, recessive, genotype, phenotype, heterozygous, probability, and the basic, all the other vocabulary related that. Now, meiosis and mitosis is, as you know, mitosis has 5 steps, and they are part of this whole cycle. Meiosis has 10 steps. Meiosis, like you see in this picture, which is like two mitosis back to back, right? As you see, they are labeled prophase 1, prophase 2, metaphase 1, metaphase 2, anaphase 1, anaphase 2, telophase 1, telophase 2, cytokinesis, cytokinesis. Eventually, the final products are 4 haploid daughter cells, and they are unique. They are not identical to each other, or also they are not identical to parents. So they are unique and they are gametes. And parent diploid, then they are actually haploid. So what meiosis is doing here, halving the chromosome number. So make sure you actually go through each of these meiosis steps. Um, and also if you see this, look at the chromosomes, right? Where are the chromosomes? And then so just following the chromosomes, you can actually tell which step of the meiosis it is. So remember, meiosis works on only special cells or gametes. Also, we see them in offsprings, parents. They actually inherit the chromosomes through the meiosis. And the fertilization again goes and combines them again, making new diploid cells. So also in plants, haploid spores in gametes exist. And there is something called gametophyte, which is one end haploid, and sporophyte, two end diploid. And there is always genetic variation, which is very, very important. So to just recap, uh, we basically in this module 3, we cover mitosis, meiosis, Mendelian genetics, chi-square test. So these 4 items. So you can go and check the PowerPoint slides in your canvas and look at some more details on these. Also, you can look at your BSC 1010 lab manual, these specific pages, chapters 8, 9, 11, and 13, and also your notes from the labs that you actually attended and you did the labs. So thanks for watching, and please feel free to like and subscribe. See you in next video. Happy studying.